Scientists had one simple rule for ancient DNA. The closer you live to where a fossil was found, the more of its DNA you should carry today. The farther away, the less. A research team decided to test that rule. They took DNA from a tiny fossil pulled out of a freezing cave in Siberia and compared it against the DNA of people living all over the world today. When the results came back, the lead scientist read them twice, then sat quietly for a long moment. The rule didn't hold, not even close. Just how badly it broke doesn't come clear until later in the story, and it's a bigger break than you'd guess. This is the story of the Denisovans, a lost branch of the human family tree we still don't have a face for. Somehow, they left more of their DNA inside people on Pacific Islands than inside anyone who lives near the cave where they were first found. Every clue in this story is written into DNA, chemical damage, genes still working inside living bodies today. It's a code nobody can fake, so subscribe now and follow it with us from the start. Before 2010, scientists thought they understood ancient Asia and Europe pretty well. Two old human relatives held two territories. Neanderthals lived in Europe and Western Asia. Homo erectus lived in East and South Asia. Two groups, two homelands. Simple. Most people have never even heard the word Denisovans. That's because this branch of the family tree didn't have a name yet. Scientists found it completely by accident. And here's the problem with that neat two-group picture. It was built almost entirely from bones, but Denisovan bones are almost impossible to find. All scientists have is one tiny pinky bone, a few teeth, one jawbone, and a handful of bone chips, small enough to fit inside a shoebox. There is no skull. Still, after more than 15 years of searching, there is no skull. That's why this whole investigation runs on DNA, not bones. Everything we know about this ancient group comes from tiny traces it left behind. Some of that DNA sits inside old bones. Some of it sits inside people who are alive right now, thousands of miles from where any of those bones were found. In 2008, a team dug up a small finger bone from Denisova Cave in the Altai Mountains of southern Siberia. In 2010, scientists tested that bone. The DNA result came back strange, very strange. A scientist named Svante Pabo was working in a very clean lab in Germany. His computer compared this ancient DNA to modern human DNA, letter by letter. It found 385 differences. For comparison, Neanderthal DNA usually differs from ours by about 202 spots. This new bone was almost twice as different as that. Pebo didn't believe it at first. He thought it might be a mistake. Maybe the sample got dirty, maybe there was a bug in the computer program, so he told his team to stop everything and test it again, from scratch. They did. The second test came back exactly the same. They had found a totally new branch of the human family, one that split off from the rest of our family tree roughly 1 million years ago. Science didn't even have a name for it yet. Then, in 2019, came another twist. A jawbone had been sitting in a museum since 1980, ever since a monk found it in a cave in Tibet. Scientists finally tested it using a new method that reads old proteins instead of DNA. It was Denisovans, too. It turned up high on the Tibetan plateau, more than 3,000 meters above sea level. And it was at least 160,000 years old, more than 100,000 years before modern humans are known to have lived there. The old tidy picture of Siberia-only Denisovans had already started to crack. Nobody yet knew how badly. At the same time, on the other side of the world, another scientist was chasing this mystery in a different way. His name was David Reich, and he worked at Harvard. He wasn't looking at old bones. He was looking at spreadsheets full of DNA data from people alive today. He used a math test called the D statistic. It's a way to search for tiny traces of ancient DNA hiding inside modern people's genes. Reich expected a boring result. If Denisovans only ever lived in that one cold cave in Siberia, then nearby people in Asia should carry a small trace of their DNA, and people far away should carry almost none. He ran the numbers. Chinese and European people showed almost no Denisovans' DNA at all, just like he expected. Then, he checked people from Papua New Guinea and nearby Pacific islands. These are tropical places, thousands of miles from Siberia. No Denisovans fossil has ever turned up anywhere close to there. The result made no sense. 4 to 6% of their entire DNA was Denisovans. He checked it again and again, still sure it had to be a mistake, maybe a software bug, maybe a bad sample. He didn't publish the result for months, but the number never changed, no matter how many times his team tested it. A tiny fossil from a freezing Siberian cave had left its biggest genetic mark in people who lived near the equator, people who had never been anywhere close to that cave. In 2010, nobody could explain how that happened. More than 10 years later, scientists are still arguing about the exact path this DNA took to get there. To answer this fairly, scientists needed better tools, so they built a whole toolkit from scratch. The D statistic was just the start. Other math tests checked how often ancient DNA mixed into modern people, and roughly when it happened. A scientist named Matthias Meyer built a new lab method in 2012. It let scientists read DNA from a tiny scrap of old crumbling bone, less than the weight of a grain of sand. Every step of this process exists for one reason: to prove the DNA is real, not contamination from a lab worker's skin or hair. So scientists handle every sample with extreme care. They work in super clean rooms with filtered air. They run extra tests to catch any sign of modern DNA sneaking in. Real ancient DNA carries a kind of chemical damage that builds up slowly over tens of thousands of years. Modern DNA does not have this damage. That is how scientists tell the difference. Even with all these tools, there is one thing scientists still cannot do. They cannot rebuild a Denisovan face or a full skeleton or a voice. All they have is the chemistry left behind. In 2022, Svante Pääbo won the Nobel Prize for exactly this work, reading the DNA of ancient extinct human relatives. So scientists set out to answer one big question: how much Denisovan DNA is inside people today, and where is it hiding? The plan was simple. Compare DNA from people all over the world against the ancient Denisovan genome and see where the matches show up. If Denisovans really had only lived in that one Siberian cave, the pattern should be simple too. The most DNA should show up near the cave, then fade out the farther you go. In December 2010, Reich's team published their results. People from Papua New Guinea, Aboriginal Australia, and nearby islands carried 4 to 6% Denisovan DNA, a huge amount. People in East Asia, South Asia, and Europe carried almost none. Then, in 2021, another team dug even deeper. They studied 118 different groups of people across the Philippines. One group stood out, the Ida Magbukon, who live on the island of Luzon. The Ida Magbukon carry 5 to 6% Denisovan DNA, higher than anywhere else on Earth, even higher than Papua New Guinea. Out of every group of people alive today, a small community on one island in the Philippines carries the most DNA from a hominin discovered in frozen Siberia. A hominin, in case you're wondering, just means an ancient human or one of our close extinct relatives. Stop and think about that for a second. Remember the rule from the start of this story: closer to the fossil should mean more of its DNA. Here's what that rule actually looks like with real numbers on it. Denisova Cave at 51 degrees north, Luzon near the equator, over 7,000 kilometers apart. So how does an ancient human relative, known mainly from one cold cave, end up leaving its biggest DNA trace on a warm island on the far side of the planet? The first thing scientists checked was the boring explanation. Could this just be a mistake, modern DNA accidentally mixed into the sample? No. This is the same chemical damage test already covered, and it clears this DNA too. But there's more proof here. The Denisovan DNA inside people today shows up in short, broken pieces, not long, clean strands. That's exactly what you'd expect after DNA has passed down through thousands of generations. It's old DNA that formed slowly over time. Then, something even stranger turned up. Scientists checked people who live in Central Asia today, right near the Altai Mountains, closer to Denisovan Cave than anyone else on Earth. Those people carry only about 0.1 to 0.2% Denisovan DNA. Compare that to the Philippines. It's a tiny amount, even smaller than what shows up in East Asia, which sits much farther away. The people who live closest to the only known Denisovan cave have almost none of this DNA. The people who live farthest away have the most. That is the exact opposite of what you would expect. Once scientists ruled out lab mistakes, only one real answer was left: geography. Denisovans never stayed locked inside one Siberian cave. Their true range must have stretched for thousands of kilometers across East Asia, Southeast Asia, and a huge stretch of land that is now underwater, called Sundaland. Here is the likely chain of events. Ancient humans left Africa and traveled along the southern coast of Asia. Somewhere in tropical Asia, they met Denisovans and had children with them, long before any of these people ever set foot near the Altai Mountains. Later, some of their children's children crossed into what is now Australia and New Guinea, roughly 50,000 to 60,000 years ago, carrying that inherited Denisovan DNA along with them. The cave in Siberia was probably never the center of the Denisovan world. It was more likely just the cold edge of it. The real center of their world was probably far to the south. If this were the only clue, it might just be a strange coincidence. But it is not the only clue. In 2014, another team found something separate and just as strong. It has nothing to do with percentages. It is about biology that simply cannot be faked. People living in Tibet today carry a special gene called EPAS1. It lets them live safely more than 4,000 meters above sea level, where the air is thin. Normally, thin air forces your body to make too many red blood cells, which makes your blood dangerously thick. This Tibetan gene stops that from happening. Scientists compared this gene to the Denisovan genome. It was an exact match. A gene that still works inside living people today came straight from Denisovan ancestors. Nobody can fake a working gene by accident, and nobody can fake that same exact match across an entire population. There is a second kind of proof, and it doesn't even need a bone. In 2020, a team pulled Denisovan DNA straight out of dirt on a cave floor in Tibet. No skeleton, no tooth, no fossil of any kind, just soil. That soil held layers of DNA from 3 different time periods, roughly 100,000 years ago, 60,000 years ago, and maybe even 45,000 years ago. Later research explained why this works. Tiny bits of DNA stick tightly to small mineral grains in the soil. They stay locked in place layer by layer and do not drift up or down over time. Modern contamination spreads messily across a whole site instead. Clean, layered ancient DNA is something contamination simply cannot copy. Then came proof of a completely different kind, this time about bone shape, not DNA at all. In 2022, scientists found a single tooth in a cave in Laos, a molar nicknamed Tom Ngu Hao 2, dating back 164,000 to 131,000 years. What proved this tooth was Denisovan was not DNA. It was the tooth's inner structure, the spot where the hard outer layer meets the softer inner layer. That structure matched a jawbone already known to be Denisovan, found in Tibet. You can measure a shape like that. You cannot fake it after the fact. Denisovans, it turned out, were living in tropical rainforests too, not only in cold Siberia and high-altitude Tibet. One more piece of evidence turns out to be the strangest of all, because it comes from what is missing, not from what is there. Across the human genome today, huge stretches carry almost zero Denisovan DNA. This is especially true on the X chromosome and in genes tied to having children. In 2016, scientists explained why. When a Denisovan father and a human mother had sons, those sons were often less fertile. Over thousands of generations, evolution slowly erased that part of their DNA. But other parts of Denisovan DNA stuck around for a good reason. They are doing useful jobs. One gene helps modern Arctic people, like the Inuit, handle extreme cold. Other genes help people in the tropics fight off local diseases. The parts of Denisovan DNA that disappeared and the parts that survived both make sense. That is exactly what real evolution looks like. For a while, one simple theory seemed to explain everything. Maybe Denisovans mixed with humans just one time, somewhere in Siberia or Central Asia. Then, that one mixed group slowly spread south, all the way to Australia and the Philippines. They carried the same dose of Denisovan DNA the whole way. It's a clean, simple story, just one meeting to explain. But the DNA itself rules this out. Scientists compared the exact bits of Denisovan DNA carried by different groups of people today. The bits are not the same. The Denisovan DNA inside the Ida Magbukon traces back to a group that split off from the Siberian Denisovans over 200,000 years ago. And that is not the whole story. Scientists found signs of at least two separate waves of mixing in this region. One wave reached Papuans, Australians, and Philippine groups broadly. A second, smaller wave reached only the ancestors of the Ida Magbukon. It came from a different Denisovan group entirely. One single meeting in one place cannot create many different DNA patterns spread across different tropical groups. Whatever happened here, it happened more than once, in more than one place. There's a second theory worth checking too. What if this whole signal is just a mistake in the math? What if it comes from old, mixed-up population patterns deep inside Africa, ones that just happen to look like Denisovan mixing? Scientists tested this directly, using several math tools built to catch exactly that kind of trick. It did not hold up. Old African population patterns cannot explain why this signal shows up specifically in Oceania and the Philippines, while staying flat everywhere in Africa, Europe, and the Middle East. So here's what the evidence adds up to. The chemistry of this DNA cannot be faked or forged. Working genes from Denisovans are still doing real jobs inside living people's bodies today. A genetic trace was pulled straight out of cave dirt, with no bones required at all. Bone structure survives inside a single tooth from Laos, shaped in a way that only real growth could produce. Even the gaps in the DNA tell a story, thinning out exactly where you would expect and clustering exactly where you would expect. Put it all together, and the trail runs from a cave in Siberia to a jawbone in Tibet, to a tooth in Laos, to the highest Denisovan DNA levels ever recorded in a small community in the Philippines most people have never heard of. Different teams found each of these clues. They used different methods. They worked decades apart from each other, and every single one of them points in the same direction. None of this tells us what a Denisovan actually looked like. DNA can tell you a gene story. It cannot rebuild a face, a voice, or a full body. No high-quality Denisovan genome has ever been pulled from tropical Asia. A genome is the complete set of DNA in a living thing. And in tropical Asia, hot, wet, acidic soil destroys DNA fast. Even the dates scientists use come with some built-in doubt. The math behind DNA aging is not perfect. It can be off by thousands of years. Honesty has to work both ways here. Separate research found signs of a mystery ancient group hiding inside the DNA of some West African people today. But there is no solid published proof that this group is actually Denisovan. Folding that finding into this story would be exactly the kind of shortcut this investigation has tried hard to avoid. Almost everything scientists know about Denisovans living south of Siberia comes from DNA signals inside living people, plus a small handful of teeth and jaw pieces. Here is what all this evidence really shows. An entire branch of the human family lived across a huge range of places, freezing caves, high mountains, tropical islands. It left DNA proof of itself almost everywhere, except in the one place people would actually recognize it: its own face. Some scientists now wonder about other mystery fossils, like Homo luzonensis in the Philippines and Homo floresiensis in Indonesia. Could these actually be southern Denisovans built for life on islands? Nobody has proven that yet. Maybe nobody ever will. If you made it this far, you now know more about where Denisovan DNA turned up than almost anyone who has heard the name Denisovan at all. This case is still wide open. Scientists are testing new soil and new groups of people right now. So hit subscribe and turn on the bell. The next clue could rewrite this whole map again. And it just might be the clue that finally gives this story a face.